Okay, good morning, everybody. And we're going to carry on our brief run through of the history of the science of microbiology. And I hope I make it through with my croaky voice. I'm a living petri dish as we speak. So I was telling you, I ended off at this slide here. We talked a lot about the discovery of microorganisms, how amazing it was to everybody that there were these living beings that were invisible. And we saw how early microbiologists worked assiduously to work out what exactly they did and began to associate them, for example, with certain well-known processes such as fermentation, but then also with disease processes. And that the work of Pasteur and Koch was particularly important. In actual fact, the association of microorganisms with disease had been made before, but in an inadvertent way. What I mean by that is that it had become realized that in certain diseases, in certain disease processes, there were systems where it was obvious that disease was being transmitted from one person to another. This was quite well known, but the mechanism was completely not known, was completely unknown. And this is particularly true of this terrible disease of smallpox. So it was well known that, for example, if somebody went down with smallpox in a household, other people in the household would very rapidly go down with the disease as well. The same thing was true of various other diseases, and it was known that the diseases spread from one person to another. The mechanism, completely unknown. But there were a lot of peripheral observations that were made around this, even from ancient times. So we'll take the disease of smallpox as an example. This is somebody suffering from smallpox. It's a terrible disease. It's a viral disease, very, very contagious. And it results in a whole spectrum of disease. Some people could have it relatively mildly. But most people, it was severe, and it had a very high mortality rate. People were absolutely terrified of smallpox, not just because it caused this illness and possibly death, but because of this, the skin became covered with these pustules and these deep-seated pox. This is the actual pox. And these could result in hideous disfigurement. Loss of nose, loss of ears, hideous disfigurements of the face and the body, with terrible scarring if the person did in fact recover from the disease. So smallpox was a disease which was regarded as being a plague disease. It would run through populations periodically and result in very, very high mortalities. Very easily identified, of course, because of its spectacular characteristic poxes. So there were a couple of peripheral observations made about smallpox from very early on. The first was that it spread. The second was it spread to people close by more easily than it spread to people further away. Evidence of its contagion. The third was that if you suffered from smallpox and recovered, you would never again suffer from smallpox. Even in a severe outbreak, people who had suffered even a mild smallpox episode at some time in their lives were immune to the disease. And this was of tremendous interest. In ancient China, and also in early Arab doctors had made this observation and had developed a system whereby they would go to people who were recovering from smallpox. And they would break some of the pustules, and they would extract pus and fluid from it. They would dry it on a needle or keep a store of the dried material. And they would then take that and go and scratch somebody with the needle with live smallpox virus. Of course, they didn't know the virus, anything about viruses, but all they knew was that this material could confer the illness. But if you took it from somebody who was recovering from smallpox, then the chances were that the person that you scratched with the needle would suffer a milder version of the disease, and in most cases would survive. There was still a high risk. The risk of death was about 10%, which is high. But the risk of death was very, very much higher, more than 50%, if you contracted the disease naturally. Now, this was a system which was referred to as variolation. Variolation, because the Latin name for this disease was variola. And variolation was a system whereby you took material from one of these pox, from somebody it was very important that the person actually be obviously recovering from the disease. Go scratch it into somebody else, and they would get the disease, but the chances were great that they would only have a mild dose, and they would recover, and thereafter be completely immune for the rest of their lives. So this was well known in areas where it was done. And in fact, there was a British diplomat, Lord Montagu. He and his wife were ambassadors to Turkey. And there was a European-wide epidemic of smallpox. And she heard, this lady, she heard about the system of variolation. And she got a doctor, a Turkish doctor, to variolate herself and her family, children. And they all survived. They got mild dose of smallpox, but they survived. And they then went back to London. And after they arrived back in London, the smallpox epidemic arrived. And her family were completely immune. And she publicized the system of variolation. It never achieved greater popularity because of the likelihood, the 10% likelihood that you could actually get severe illness and die. But nonetheless, some people did it. And it's where people survived, it certainly worked. And it was a positive demonstration that immunity to a disease could be acquired and that it could be artificially induced. The problem was the danger associated with doing it this way through variolation. But this gentleman, Edward Jenner, formalized another system. It was based on the same kind of idea, excepting that Jenner heard that there was this, well, it was actually well known that milkmaids did not get smallpox. And when he inquired further into this phenomenon, he discovered that it was not only that milkmaids didn't get smallpox, but that milkmaids who had suffered from another disease called Karlpox did not get smallpox. Now, Karlpox is a related virus. It infects cows. It does not cause serious illness, but it's well known, and it produces blisters on the udders and on the nipples. And it can be acquired by close contact with human beings. So milkmaids would milk the cows with cowpox, and they would develop blisters on their fingers And that was all that happened to them. They would just get these blisters on their fingers. It would eventually go away. It was just an occupation. They regarded it as an occupational hazard of being a milkmaid, but it made them completely immune to smallpox. It was not the smallpox virus. It was a pox virus, but not the smallpox virus, and it did not cause serious illness in human beings. Jenner heard about this, and he developed a system of collecting cowpox exudate instead of smallpox exudate, and inoculating people with the cowpox virus. And they would then become immune to smallpox perfectly successfully with no illness, with no illness on their own part. And he named that system vaccination because vacca is the Latin word for cow. That is where we get our term vaccination, now so well known to us all. This is where the word vaccination comes from. And notice, it's really important to notice the difference between the old system of variolation, which took actual smallpox virus and inoculated people with it, and vaccination, which used a harmless, different virus. Now, we now understand, because we have some understanding about the immune system, we now understand that our immune system responds to cowpox virus, even though it doesn't cause serious illness. Our immune system recognizes it and develops a system of antibodies and memory of the virus. And when it sees smallpox virus, the smallpox virus looks so similar to the cowpox virus that it causes the immune system to respond as if it was cowpox virus. If ever the immune system, once you've had the cowpox vaccination, if ever your immune system sees smallpox virus, it responds to the smallpox virus as if it was cowpox virus, and it wipes it out. And you are effectively immune. And now Jenner's system, the people were immune with minimal risk because the cowpox virus couldn't cause serious illness in human beings. And that came to be referred to as immunity. The people acquired immunity to a particular disease. Look at the date. This is the end of the 1700s. And so Pasteur and Koch and everybody, they're going to be working within 40 years or so of Jenner's publication. I won't say publication, but Jenner embarked on a great publicity campaign in order to publicize his methods. And it's resulted in very, very successful campaigns against smallpox, even where they had no understanding of the actual mechanisms that they were utilizing. But Pasteur was very interested in this. And he knew all about variolation. And he kind of wondered, why was it that variolation using the actual smallpox virus did work? And when you did that, when you variolated somebody with smallpox virus from somebody who had been recovering from smallpox, they had a much lower chance of getting serious illness than if they had the wild virus. And he reasoned that what had happened was somebody who suffered from smallpox, they went through their illness and their own body began to fight the virus. And the mechanism, he of course had no understanding of the mechanisms whereby the body was fighting the virus. But he reasoned that what happened was the virus became weaker and weaker and weaker. Its ability to cause illness got less and less and less as the illness proceeded. And of course, the immune system we now understand overcame the virus with antibodies and everything else. The virus became weaker and weaker until it died out. He used that reasoning and said, well, how about if I take disease-causing organisms and I pass them through a series of experimental animals So what he did was he primarily used rabbits, sometimes sheep, but he used rabbits mostly. He would infect a rabbit with a disease, and he would then let the disease progress. Often at the first rabbits, of course, would very soon become extremely ill and then die, but before they die, he would extract blood or whatever and inject another rabbit, and then inject another rabbit, and then inject another rabbit. This is a system called passage, and it is still used in vaccine development. As the disease-causing organism, not necessarily a virus, it works with bacteria too. As the disease-causing organism is passaged through these different subjects, it becomes weaker and weaker. Its ability, its pathogenicity is reduced and reduced and reduced, until in the end you have the living organism. You can extract it at the end, but it can no longer cause disease, or at least can no longer cause serious disease. And by doing this, Pasteur developed vaccination systems for a number of different diseases. And he called what was happening, he called it attenuation. It was the principle of attenuation, reducing the pathogenicity of an organism by passage through a series of subjects until such time as the organism became non-pathogenic, but would still excite immunity if it was given. So he did this for a number of diseases. The one that he was most famous for initially was chicken cholera. It's actually not cholera, but it is a diarrheal disease in chickens, which is devastating for chicken farmers. And he successfully developed a vaccine for chicken cholera and became very famous and probably quite rich because of it. The next one that he also worked on anthrax and developed a quite successful. Anthrax is a very difficult organism to work with, extremely, extremely dangerous. But he developed a good vaccine for anthrax, especially for cattle. But his most famous one was for the disease of rabies. Now, rabies is a terrible, terrible, terrible disease. It's still quite common here in the United States, continental Europe, and all through Africa and Asia. It occurs quite widely. And rabies is a virus which can infect virtually any mammal. And it causes a neurological disease. And the classic example is the mad dog, of course. As the neurological symptoms progress, it induces behavioral changes. And one of the things that it does is it causes animals that are infected with it to begin to salivate, because the virus is transmitted largely through saliva. And it also induces animals to become extremely ferocious and to attack. They will attack inanimate objects and living things and everything. And of course, this is another adaptation, because this is a way that the virus can be spread from subject to subject. So rabies was well recognized. It was absolutely dreaded, because if you were bitten by a rabid animal, the infectivity is very, very high. And once you begin to show symptoms of rabies, mortality is 100%. There are 1 or 2 people who have been rescued from rabies in recent years, but it is extremely rare, even in modern times, for somebody who's symptomatic for rabies to survive the illness. So since ancient times, rabies has been a dreaded disease. And in France, this was true, as it was anywhere else. And Pasteur worked on rabies, He was extremely scared, though. He developed a system whereby he passaged it through rabbits and ended up with a solution which seemed to induce immunity in rabbits without inducing illness. But he was terrified to use it. He did test it on himself, but he was terrified because just in case it went wrong, because he knew it was living virus. So he didn't ever want to use it in human subjects. But in the south of France, a woman and her son were out walking around, and the son was attacked by a rabid dog and very severely bitten by this dog. And it was obviously rabies, and it was obvious that this boy was probably going to get rabies and die. And she had heard about this wonderful Dr. Pasteur in Paris, and she gathered whatever money she had together, and she took her son to Paris and asked Pasteur to do something to save her son. And all he had was the vaccine that he had developed from his rabbits. And he decided, yes, that he would, in fact, vaccinate this boy. Now, here's the principle that he's using. The boy has already been infected with the virus, but it was known that rabies developed extremely slowly. What actually happens is the virus enters nerve cells in the region where the infection takes place, where the bite, for example. And it then slowly makes its way up the nerves into the central nervous system, and then develops in the central nervous system as the fulminant illness that you observe. That takes a long time. It takes, depending on how close to the brain the bite is, it can take days to weeks for the symptoms to develop. Pasteur reasoned that if he gave this boy the vaccine, he could induce the immunity fast enough that it would overtake the infection. And that is exactly what happened. This was the first instance where somebody known to have been infected with rabies was actually rescued from the illness by vaccination. It was actually, he actually had to have several vaccinations. He did it over a period of time, but it worked perfectly successfully, and was the one thing, this was one incident that made Pasteur more famous than anything else. The story, it was regarded as, he was regarded as being a miracle worker because of this. There's a nice little end to the tale, and that is that this boy then, when he grew up, moved to Paris and became Pasteur's laboratory assistant, and actually ended up running his laboratory in Paris. And in fact, during the Franco-Prussian War in the 1800s, Pasteur fled Paris, had to flee Paris, and the boy refused, well, not then a boy, but he was being a man, refused to leave the laboratory. And he actually prevented Prussian soldiers from entering and destroying the laboratory. They were busy wreaking havoc all over Paris, and he protected the laboratory. And when Pasteur eventually returned, his laboratory was pristine and had been kept by this chap. He worked for Pasteur for the rest of his life. So that's the Pasteur, along with everything else, became extremely famous for developing these systems of vaccination. Again, he did it with only minimal understanding about the processes of infection and of immunity. He did it empirically. He did it by observing outcome without understanding mechanism, but nonetheless developed these systems quite successfully. So this was certainly for most workers, for most microbiologists and bacteriologists, this was a wonderful development. the idea that we could actually prevent disease, and we could do it now by various mechanisms. First of all, remember the work of Lister, Semmelweis, these people who showed that ordinary things like washing your hands, observing simple hygiene, and this kind of thing could limit the spread of disease from one person to another, or from one source to a human. That is the first. The second thing, Pasteur, Jenner, these people showed that for several diseases, not, it has to be said, not for all diseases by any means, but for several diseases, systems of vaccination could be developed, which would arm people with immunity to disease, which even if it didn't prevent the infection altogether, in many instances would lead to people simply having mild illness if they were ever exposed. These simple developments began, believe it or not, to have an enormous effect upon the population, upon the human population. From this time, we begin to trace the diminishing importance of infectious diseases in human mortality. But the third thing which everybody wanted to reach was to get to a point where when people were ill, they could be given substances which would mitigate the illness or eliminate the organisms that cause the illness. And this was the desire to develop chemotherapies for treatment of illness. And when, in fact, this began to be done, then this ushered in a second golden age of microbiology, which was the era where we began to actually combat infectious disease by striking at the organism that causes the disease, even after symptoms have arisen. And the aim of the chemotherapy was to develop drugs which would affect the microorganism that caused the illness without or with minimally affecting the host. It's no good to, you can, if you want to, if somebody's infected with somebody, you can saturate them with arsenic, right? It'll kill the microorganism, but it'll also kill the person who has the microorganism. Now, it has to be said that up until this time, the second golden age, there was virtually nothing. It's an extraordinary thing to think about, but until the 1900s, there was virtually no specific drugs of this nature. And up until that time, medicine and doctoring had consisted of attempts to treat illness with the most extraordinary range of treatments, all the way from draining people's blood to balance their humors, to giving them extremely poisonous substances like arsenic, lead oxides, mercury, which is very, very commonly used to try and treat infectious disease, etc., etc., etc. In actual fact, medical personnel were responsible for more deaths than the microorganisms themselves. There was very little that could be done. There was one substance which had been known for centuries, but became especially popular during 1700s and 1800s. And this is the substance quinine. And quinine is a naturally occurring substance. It was discovered in South America, where Indian tribes in South America were observed using it to treat malaria and other parasitic diseases. An extremely bitter substance extracted from the bark of the quinine tree. But this was one of the very few substances, drugs available with a specific action. It was especially effective for malaria and for preventing malaria as well as for treating it, although not 100% successfully. Still, it was something. Quinine was one of the very few things available. Other than that, it was mostly attempts to use known poisons like mercuric oxide and these sorts of things in low enough doses that it didn't kill the patient, but hopefully that it killed them. There was one disease which drove this search for this magic bullet. The magic bullet is a term which was derived by somebody called Ehrlich, another bacteriologist, microbiologist. And the idea of the magic bullet is that you get a substance which kills only the microorganism, doesn't kill any other cells, doesn't affect the host. The disease that everybody was worried about, primarily that drove this, was syphilis. And syphilis was actually a relatively new disease in European populations. It had entered probably in the 11 or 1200s into the population and had run rampant, and is a disease which causes terrible consequences. Syphilis has various stages. It has a primary stage, which is quite easy to recognize. It's caused by a bacterium, which is a spirochete. The first stage is a sore, which is quite easily seen, especially in men. It then appears to heal and disappear, but in fact goes into a secondary stage where it begins to multiply throughout the body. And it then can appear anywhere from years to decades later as a tertiary stage with terrible consequences, huge disfigurements of facial features, for example, neurological damage, dementias also. And so syphilis was a dreaded disease. And often people knew that they had been infected with syphilis because the primary syphilis, the sore, is quite easy to recognize. So this drove the attempt to find some drug cure, especially for syphilis. And it led into the development of many of these heavy metal substances, which were then used to try and treat it. In more modern times, towards the end of the 19th century and then in the beginning of the 20th century, much more specific drugs began to be developed. We recognized they are all antibiotics. Let's just make that clear. They're all antibiotics in the sense that they kill living things, right? But we really recognize two groups of these drugs. The first group are the entirely synthetic drugs. They're substances which do not occur in nature, but they are synthesized and have action against microorganisms. When we in modern times talk about antibiotics, the term is a little bit loose, but the term antibiotics really specifically refers to chemicals that are produced by bacteria and fungi principally that inhibit or kill other microbes. So this needs some explanation. It sounds like a tautology, right? Here we have bacteria and fungi especially, which are producing substances which are antibiotics. They kill microorganisms. Why would a microorganism produce a substance which kills microorganisms? The answer lies in the fact you need to remember that these microorganisms are competing with themselves, with one another the whole time for limited resources, limited nutrients, and especially limited space. Many bacteria, for example, need to be attached to a surface or part of a biofilm, a film of bacteria, in order to function properly. So they compete with one another for space to grow. They also compete with one another for nutrients and everything that is in the environment. So if a bacterium or a fungus can produce a substance which will kill its competitors, its competing bacteria and competing fungi, then it will have that space to itself and all that nutrient to itself. And that is the principle of antibiotic production. And bacteria and fungi especially produce a wide range of these substances, which have very specific action against other microorganisms which might compete with the producer. So those are the true antibiotics. So the synthetic drugs, you will still hear people refer to them as antibiotics, simply that they kill living things. But truly, antibiotics are those substances produced by living organisms. So I've already told you, Paul Ehrlich speculated about the development of these magic bullets that would harm a pathogen without harming a host. And he worked assiduously to try and do this. He went actually to the old drugs that had been used, especially those, especially the use of arsenic. Arsenic is a heavy metal which is extremely, extremely poisonous. But he developed a synthetic drug based on arsenic, and that was called salvarsan. And in 1910, this was, and here was where he did this specifically to treat syphilis. And salvarsan was used for decades as a treatment for syphilis, and it was quite successful. And became very widely used. So salvarsan really was the first synthetic drug with specific activity against bacteria. Remember that syphilis is caused by a spirochete bacterium. Now, in the 90s, all of this research continued. People were desperately looking for drugs with specific activity against microorganisms. And in the 1930s in Germany, sulfonamides were first synthesized, and they were produced actually from pigments, that were being used in the dyeing industry and things. And they are true antibiotics in that they kill living. They have specific activity against living organisms. And they were extremely popular because they were active against many pathogenic organisms, many pathogenic bacterial pathogens. But unfortunately, the sulfonamides also had some really bad side effects. They're actually very toxic. And in addition, this was the first instance where people began to notice that bacteria could develop resistance. That where at the start of their usage, the sulfonamide might be quite successful in combating a bacterial infection. If it was continued to be used, and especially if it continued to be used under certain circumstances, in a hospital, for example, where there was ongoing reinfection with the same microorganism and ongoing usage of the sulfonamide, eventually the bacteria would become resistant and the sulfonamides were no longer effective. They would then try to modify the sulfonamide a little bit so that it would reacquire its effectiveness, but soon resistance would develop. Nonetheless, sulfonamides were quite successful, and especially against the great scourge of this time, which was tuberculosis. It was one of the first, the sulfonamides were one of the first drugs which could be successfully used in treatment of tuberculosis. Then, early in the 20th century, 1928. Excuse me, I'm starting to lose my voice. This very, very famous bacteriologist, Alexander Fleming, discovered the first true antibiotic, the first biologically derived antibiotic. And he did it completely by accident because he went on holiday. And this is the way it happened. He was busy running experiments. He was actually working on Staphylococcus aureus, and he was growing it on plates. Had many of them growing on plates all over his lab. Here they are here. These are colonies of the Staph aureus. He went away on holiday, and when he came back, he had left plates out. The very bad practice in microbiology. You never leave plates just sitting around on. But for the very good reason that when you do that, you start to grow fungus. When you get fungus into your lab, it can be very difficult to eliminate fungus. And that's exactly what happened to him. Here he observed that he had plates of Staph aureus, and some of them had become infected with this fungus here. And this is the actual plate that Fleming observed, that he wrote up. And here's the this is a fungus called Penicillium. That name should ring a bell. The fungus name is Penicillium. Penicillium means little brush. And that's because they have their reproductive structures look like a little paintbrush, like this. So here it is growing here. Here's the he had originally spread this plate with Staph aureus, but he observed that the Staph aureus only grew well when it was at a distance. And if you look carefully, you'll see that there's actually almost a circle here, like that. And then inside that circle, the Staph aureus really struggled to grow. There are colonies here, but they are very small. The bacterial growth is actually strongly inhibited by the presence of penicillin, of the Penicillium. And he reasoned that what was happening was that the Penicillium was producing a substance which inhibited bacterial growth. And the substance was being released as an exoenzyme. It was being released into solution around the colony and was drifting out and was inhibiting bacterial growth all around here. And for Fleming, that was basically the end of it. He wrote up his findings, but he didn't make the great leap for many years. It took other people actually to make to look at that work and say, wait a minute, maybe we could actually do this deliberately. Maybe we can take Penicillium and culture it under circumstances where we encourage the development of this exoenzyme, and we can then use that. We can actually use that as a drug. And that is, it took a tremendously long time and a huge amount of research to purify that substance and begin to use it. And that is the substance penicillin. That is the very first true antibiotic. It is actually produced by a living organism, by the Penicillium fungus. It is released into the environment in order to outcompete bacteria, which would compete with the fungus. But it took until the 1940s. It was only during the Second World War that penicillin was tested clinically and then mass produced and began to be widely used. It was especially important in treating battle wounds, because up until that time, there was very little that could be done. They did use sulfonamides, but they had limited usage for treating infection of wounds acquired in battle. Penicillin was the first really successful antibiotic, and it was spectacularly successful. It is especially useful against gram-positive bacteria because it inhibits the growth of a cell wall of gram-positive bacteria. So that's main usage. But gram-positive bacteria are a very common pathogen. in humans. It does also have activity against gram-negative bacteria, but is much more strongly active against gram-positive bacteria. And it was spectacularly successful. It began to be used very, very, very widely throughout the world in order to treat bacterial infections and bacterial illness. It very quickly became apparent that they were ran into the same problems that other antibiotics ran into. And that was that that bacteria very rapidly developed resistance to the presence of the antibiotic. So I just want to take you back to here to try and explain something. Just have a look here. This is Fleming's original. Now, what is happening here is this is the wild fungus growing under almost wild conditions, releasing it. When you look here, you realize there's this zone of inhibition. But even look here, very, very close to the fungus itself, there are bacteria which are strongly inhibited, but they are still growing. They are still growing right up there against them. Now, here's the problem. When we treat a population with an antibiotic, what we're doing is we're actually treating a very diverse population. The bacterial population inherently is diverse. It has many different properties, even in the same species of bacteria. And some of them are more resistant to the antibiotic than others. And if you're treating with antibiotics, if any bacteria survive, what are they going to be? They're going to be the resistant bacteria. The resistant bacteria have a greater chance of surviving treatment than those that are susceptible. If any bacteria survive, then they are going to reproduce, and all of their progeny are going to be resistant to the antibiotic. And if you now come along and try to treat with the antibiotic, you're treating a population that has a much higher inherent resistance. And now, even if you treat with the antibiotic, you manage to kill off some of them, who survives? A much higher proportion of the population survives with the genes that confer resistance. This is how we develop resistant populations in bacteria. There are some other mechanisms that we'll hear about as well when we talk about bacterial genetics. But it soon became apparent that the overuse of antibiotics was in fact extremely dangerous, and it still is. It has led to the development of bacterial strains which are resistant to almost every known antibiotic and chemotherapeutic agent. And we are increasingly realizing that we're facing a crisis in medicine. We have enjoyed a honeymoon period for the last 100 years or so of antibiotic usage and chemotherapeutic usage, but the honeymoon may soon be over as all of these resistant strains begin to appear. The research into the genetics of microorganisms, it originated in this realization of how resistance was developing, but has led to what has often been referred to as the third golden age, the golden age of microbiology, which is examining the actual genetic mechanisms for bacterial life. And that's especially since the discovery or the elucidation of the structure and function of DNA, and the realization that DNA could actually be studied in terms of its structure, its sequence, but also that DNA, that the genetic structure of microorganisms could be manipulated. And these are the sciences of cellular biology and of biotechnology, which we'll talk about later in the semester. Okay, so we arrive now at the present day. When we go out, we say, okay, microbiologists, what is a well, there are many microbiologists. There are lots of people who study things which cannot easily be seen with the human eye, naked eye, who would be classified as microbiologists. But within that, there are a number of different disciplines. Bacteriology is specifically the study of bacteria. And bacteria are not the only prokaryotes, and they're certainly not the only microorganisms. So that is a specialization. The study of bacteria themselves is the study of bacteriology. Mycology is the study of fungi. Unfortunately, in this class, we're not going to talk about fungi very much. You may wonder, look, many fungi are easily visible with the naked eye. So you may wonder why a microbiologist would be studying fungi. And the answer is there are many, first of all, there are many fungi which are single-celled, so the yeasts, which are truly microorganisms. But in addition, fungi have many microscopic stages to them, to their development. The third branch of microbiology is the study of parasitology. Now, parasitology, that word is somewhat confusing, because if you think about it, we need to find a definition of what is a parasite. And a parasite, in biological definition of parasite, is an organism that lives in close association with another organism at the expense of that organism. In other words, a parasite damages its host, but does not itself make any contribution to the host. And if we think about it in that term, then bacteria really are parasites as well, because that's exactly what they do. They inhabit a host, a pathogenic bacteria. They inhabit a host. They derive all sorts of benefits from that association, but they don't confer any benefits. If they're a pathogen, they don't confer benefits to the host. So they're parasites. But we don't regard bacteria and viruses really as being parasites. Parasites are organisms which do that. They exist at the expense of their host, which are not bacteria or viruses. And the vast majority of them are, of course, then that means the vast majority are eukaryotic. They are eukaryotic microbes, and especially protozoa and parasitic worms. Those are the two major groups of parasites, and their study, the study of them, is parasitology. Now, we've talked extensively about bacteria and the prokaryotes. The prokaryotes, of course, are the bacteria and the archaea. But we need to say a word about viruses. We will discuss viruses in detail in this class. And the study of viruses is virology. Now, the existence of viruses was noted very, very early on in the whole history of microbiology. And it was discovered, especially when people were working on bacteria, which cause disease in plants. There had always been a tremendous amount of interest in bacteria causing disease in plants because of their importance in agriculture. And people working on plants often use tobacco as an experimental subject. It's a very easy plant to grow. It's a very good experimental subject in the laboratory. And they noticed that there was not only bacteria caused disease in plants. In microbiology, sometimes we need to develop media which are extremely sensitive to heat. And they still have to be, those media still have to be sterilized in order to grow the bacteria that you're interested in. This is true of many plant bacteria. They need very specialized media, and the media cannot be boiled or autoclaved. They can't be heated. To sterilize them, they have to be passed through a filter. And the filter it's a simple mechanism. The filter is small enough that it will trap any bacteria. it'll trap bacteria down to the size of 0.2 microns, for example. So you pass your medium through this very fine filter, and what comes out the other side of the filter should be sterile. It certainly has no bacteria in it. These workers noticed that sometimes when they did this, when they filtered a media like this, they still had an infectious agent present. They could still cause disease. And in fact, they discovered that they could transmit a specific disease by doing this, and was called tobacco mosaic disease. Had very specific symptoms, and they could pass it from plant to plant, even through the filter. And so they called these the infectious particles because they had no idea what they were. They called them filterable particles. And they, in 1935, were specifically identified as this structure called the virus. And the structure was revealed once electron microscopy was developed. Viruses are far too small to be seen with the naked eye, but they can be visualized with the electron microscope. And here's the tobacco mosaic virus, often just called TMV. There it is there. And this is one of the very early electron micrographs, and one of the very first electron micrographs of a virus. So the study of viruses is virology, and we will spend a little bit of time talking about virology. We'll also spend time talking about molecular genetics. Now, some people, especially molecular geneticists, like to hive themselves off and say they're part of the fourth golden age of microbiology, which is the study of microbial genetics. And this arose as soon as the Watson, Crick, and Wilkins, his name always gets left off, and everybody talks about Watson and Crick, but it's actually Watson, Crick, and Wilkins who published the original paper on DNA structure. And we'll talk some, of course, about DNA structure, but I'll simply tell you now that this DNA structure is actually rather simple. It's actually quite a simple molecule. It's a very, very big molecule. And it's all of its properties lie in the sequence of molecules that make it up. There are 4 bases that make up DNA, and they can occur in any order. But their order is extremely important because their order is a code, and it is spelling out. In actual fact, it spells out instructions for the cell, either to make something or of one particular thing or another, or to do something. Molecular biology is the study of how DNA directs protein synthesis. The most important function of DNA is to drive the synthesis of all of the proteins that make up and direct the metabolism of the cell, as you learned in your first semester of biology. Genomics is the study of an organism's genes. Of the genes are the recipe, usually either for protein or for some substance, sometimes a recipe for RNA. And the genomics is the study of those genes, and especially it leads to the science of recombinant DNA, of taking genes and manipulating them, maybe taking genes from one organism and moving them to another organism in order for that organism to do something special, something useful. And the very first instance of this being done was by this worker, Paul Berg, who took animal DNA and put it into bacteria and showed that he could get bacteria to actually produce an animal protein that they had never before produced. And this is the basis of a whole science and a whole industry of molecular genetics and of biotechnology, of using biological systems to actually do something useful. The last discipline that we will discuss in this semester is that of microbial ecology. Now, I am actually a microbial ecologist. I was a marine microbial ecologist, so this is close to my heart. This is, in fact, one of my pictures. And what you see here, this is a marine, this is a picture of a sample of marine water. And all of these little glowing dots, these are bacteria which have been stained with a fluorescent dye. And we do this in order to count them. This is a very, very, very small volume of water. The marine systems, for example, have between 10 to the 7 and 10 to the 8 bacterial cells per milliliter. Very high number of bacteria. And these are naturally occurring bacteria. These are not pathogenic bacteria. These are bacteria which are living in the environment, living off the environment in one way or another. And our entire biosphere depends upon the functions of these microorganisms. And they, for various reasons. But here's the principal one. Think of it this way, that our planet is an enclosed system. It is a unit. It receives very, very little input from outer space. We get a small amount of material comes in, but essentially all of the molecules which we have on our planet are what we have to deal with. We don't receive new supplies from anywhere. Living processes take those simple molecules and they build them into complex biomolecules, often very complex biomolecules. If we're in an enclosed system like this, with very active living systems, eventually the living systems, in theory, should take all of the simple molecules and build them all into complex biomolecules. If those complex biomolecules stayed the way they were forever, the entire system would break down. Instead, we have very complex systems of bacteria and fungi which recycle our molecules the whole time. They don't only recycle our molecules, they also, many of them are autotrophic and are able to take carbon dioxide and water and put them together and make biomolecules. So the entire functioning of our biosphere depends upon these organisms here, upon bacteria, fungi, which are free in the environment. Our attention in microbiology is often straight to the disease-causing organisms, right? Because that's what catches our attention. But in fact, far more important are the environmentally occurring bacteria, which drive all of these processes that keep our planet functioning. So we will spend some time talking about that as well. And I'll leave you there for the day, and I'll see you then on Monday.